Hello students. In today's lecture, I am going to introduce you all to human reproductive system. Let us quickly see the topics that will be covered in today's lecture. Today you will learn about sex determination, sex differentiation, and also I will describe the anatomy of male reproductive system. The pre-pubertal sexual growth involves the growth of human embryo, and this growth involves two processes, namely sex determination and sex differentiation. Sex determination in simple words means determining the sex or the gender of the growing fetus. Thus, it refers to the genotype of the fetus. The genotype is determined by the presence of sex chromosomes. Therefore, sex determination is also called as chromosomal sex differentiation. Before understanding sex determination, you must understand this that human beings have total 46 chromosomes or 23 pairs of chromosomes, amongst which 44 chromosomes are similar in both male and female, but 2 chromosomes are different. These 2 chromosomes are called as sex chromosomes and they are X and Y. So, in females, the total chromosomes will be 44 autosomes plus 2 X chromosomes, that is 44XX, and in males, the total number of chromosomes will be 44XY. Human gamete are sperms and ova, which are produced by male and female respectively. It is the fusion of these male and female gametes that leads to the development of an embryo which grows into a baby. During gametogenesis, that is spermatogenesis in males and oogenesis in females, a special type of reductional division takes place called as meiosis that converts the cells into half. Therefore, mature sperm after spermatogenesis will contain 22 pairs of autosomes and one sex chromosome, and also a mature ovum after oogenesis will contain 22 pairs of autosomes and one sex chromosome. In females, the female gametes ova will have 22 X chromosomes, and in males, the male gametes, that is sperms, half of the sperms will have 22 X and other half will have 22 Y chromosomes, and thus they are called as haploid. Here, with this diagram, you will understand better how haploid male and female gametes form a diploid zygote or an embryo. Here you see the female gametes which are formed from 44XX are divided by meiosis to form 22X, and on the right side you can see how male gametes which are half 22X and half 22Y which formed from the 44XY. Now, during fertilization process, when the sperm and ovum meets, if 22 eggs from female combines with 22 eggs from male, a female child, that is 44XX, is formed. Whereas if 22 eggs from female and 22 Y from male undergoes fertilization, a male child having 44XY chromosomal pattern will be formed. So, it is very clear that the sex or the gender of the child depends upon the male and not female. After fertilization, the somatic cells multiply immediately, and one of the X chromosomes of the somatic cells, in case of a female embryo, is made inactive. This inactive X chromosome forms a condensed mass called as bar body or sex chromatin. It can be seen most commonly in polymorphonuclear cells, that is a type of WBC, that is white blood cell, and also in buccal and vaginal mucosal layers under a microscope. The significance of bar body is that it is used to identify the genotype of a female when there is doubt. Next topic is sex differentiation. After fertilization, the normal sex differentiation in the embryo proceeds in a sequence. The stages are namely gonadal differentiation, genital differentiation, and psychological differentiation. Gonadal differentiation or gonadogenesis means the formation of the gonads, which are testes in case of male and ovaries in case of females. The gonadal sex differentiation is dependent on the genotype of embryo. What happens here is that during gonadogenesis, a urogenital ridge is formed on each side near the adrenal glands. This urogenital ridge further develops into a bipotential gonad. Bipotential gonads, also called as primordial or primitive gonad, it has the potential to develop into both male and female embryos. Thus, the bipotential gonad is identical for both the sexes till 6th week of gestation or intrauterine life. Now, when a male child is developed, there occurs testicular differentiation of the bipotential gonads, and in this, genotype plays a major role. That is, if Y chromosome is present in the embryo, then it will undergo testicular differentiation, and if it is absent, then it will undergo ovarian differentiation. Now suppose Y chromosome is present. Then at about 6th week of gestation, primitive sex cords will arise from the germinal epithelium of the medulla of the bipotential gonads. And by 9th week, Leydig cells will start secreting the male hormone testosterone, and thus under the influence of testosterone, the embryo will develop into a male fetus. Why the Y chromosome is so important to form a male child? It is because the Y chromosome contains two genes, namely SRY and MIS. SRY, or the sex determining region of the Y chromosome gene, encodes for a substance known as testis determining factor (TDF) that triggers the testicular differentiation, whereas the Mullerian duct inhibitory substance, that is MIS, inhibits the development of embryo to grow into a female fetus by inhibiting the Mullerian duct growth. Now, in ovarian differentiation, at about 10th week of intrauterine life, a female embryo starts to develop in the absence of Y chromosome and the related hormones. The primitive ovaries develop on each side from the cortical region of the bipotential gonad. The cortical cells proliferate and forms a granulosa cell layer which commits to form oocytes in future steps. The Wolffian ducts degenerate in the absence of testosterone, and around 11th to 12th week of gestation, the oogonia which is formed undergo meiotic division to form oocytes. The ovary at this stage does not secrete any hormone. Genital sex differentiation means the differentiation of the embryo's internal and external genitalia. Let us first see the differentiation of internal genitalia. The internal genital organs of both the sexes arises around 6th week of intrauterine life from a neutral sex anlagen that develops along with the bipotential gonads. The earliest form of internal genitalia for both sexes are a pair of Wolffian ducts and Mullerian ducts. By about 7th week of intrauterine life, the embryo has both of these ducts. Later, under the influence of Y gene, the Wolffian duct will grow further to develop into a male genitalia, and it regresses the Mullerian ducts. Absence of Y gene will cause the reverse. Second is the differentiation of external genitalia. The external genitalia also develops from a common anlagen, namely urogenital sinus, genital tubercle, and genital folds. These are bipotential till 8th week of intrauterine life, that is, it can develop along either male or female lines. In a genetically male fetus, the presence of hormone testosterone and dihydrotestosterone gives the male features to the fetus by 5th month of intrauterine life. And in a female fetus, the absence of testosterone and any other hormones leads to the development of the female type of the external genitalia. Next topic is disorders of sexual development. The abnormalities or disorders of sexual development can occur due to either defect in the sex chromosomes or hormonal abnormalities. The first common disorder of importance is Klinefelter syndrome. It is a chromosomal abnormality where the individual has one extra X chromosome. The pattern of chromosomes in such individual is XXY instead of XX or XY. The individuals are genetically male but shows the following features that are not present in a normal male. The individual will have poor development of testes and this will lead to infertility. Thus, these individuals will be tall and obese with normal male internal and external genitalia built. They will have gynecomastia and their secondary sexual characters are poorly developed. The second common disorder after Klinefelter syndrome is Turner syndrome. The individuals with Turner syndrome have the karyotype 44X0 or XO. Here, the Y chromosome is absent and the individual is genetically female. Turner syndrome individuals will have ovarian dysgenesis as two X chromosomes are required for the development of ovaries. They will have delayed puberty, amenorrhea and infertility. They will suffer from mental retardation and issues related to heart. They are dwarfs with loose folds present all over the neck region. They have small jaw and drooping of upper eyelids known as ptosis. Next topic is anatomy of male reproductive system. The male reproductive system comprises of the internal and external genital organs which are gonads or primary male sex glands, accessory sex glands and ducts of the male reproductive system. Now, firstly, the gonads in males are a pair of testes. The main functions of the testes are to produce sperm and to secrete male hormone testosterone. Next, the accessory sex glands are seminal vesicles, bulbourethral glands, and prostate gland. Seminal vesicles secrete a thick alkaline fluid, whereas the bulbourethral glands secretes mucus like secretin. The prostate gland secretes a thin milky fluid and all these secretions are present in the semen. The ducts of the male reproductive system includes the epididymis, the ductus deferens, ejaculatory ducts and urethra. The sperms can remain viable in the epididymis for almost 1 month where they get nourishment and they mature. The vas deferens is the continuation of the tail of the epididymis that ends in the seminal vesicle. The ejaculatory duct arises from the union of the vas deferens and seminal vesicle and opens into the prostate urethra. And ultimately the urethra provides an exit for the semen. This is a diagram showing the male reproductive system. This is the testis that consist of many seminiferous tubules and the vas deferens arises from the testis and it opens up at the tip of the penis. The secretions of all the glands are carried by the ejaculatory duct. The supporting structures of the male reproductive system are spermatic cord, scrotum and penis. The spermatic cord helps to suspend the testis into the scrotum. The scrotum is a fibromuscular sac that houses testis. It is maintained at a temperature of 32 degrees Celsius physiologically so that normal spermatogenesis can occur. The penis is the male copulatory organ and is made up of 3 erectile tissue bodies, namely corpora cavernosa, which are 2 in number, and 1 corpus spongiosum. Testes are ovoid bodies suspended by spermatic cords in the scrotum. In an adult male, the average weight of each testis is around 25 grams, which decreases with old age. It is made up of three layers or coverings from interior to the exterior. The innermost layer is tunica vasculosa, made up of loose connective tissues. The next layer, tunica albuginea, is called as the capsule of the testis, and it consists of dense collagen and elastic fibers. And the outermost layer, tunica vaginalis, is made up of mesothelial cells. This is a diagram of testis showing the seminiferous tubules that are present in compartments. Then the epididymis that continues into the vas deferens. You can also see the three layers of the testis, tunica vasculosa, then tunica albuginea, and the outermost tunica vaginalis layer. The entire content is hanging by the spermatic cord inside the scrotal sac. The arterial blood supply to the testis comes from testicular arteries, and the venous blood is drained by the testicular veins that joins to form a special plexus called as pampiniform plexus. The significance of this plexus is that it helps in the thermoregulation of the testis, that is, it maintains the constant low temperature of the testis compared to other body parts. The nerve supply to testis arises as a testicular plexus of nerves from the autonomic nervous system. Each testis is divided into many lobules by the fibrous septa, which project from the mediastinal testis into the tunica albuginea. Each lobule is roughly conical in shape. Each lobule of the testis consists of two compartments, seminiferous tubular compartment and interstitial compartment. The seminiferous tubular compartment of each lobule of the testis contains about 2 to 3 seminiferous tubules. It consists about 80 to 90% of the volume of the testis. See here, these are the seminiferous tubules present inside the lobes. Each seminiferous tubule consists of two parts, a convoluted part and a straight part. The convoluted part forms the loops and continues as two straight ends. Near the apex of the lobules, the straight ends join one another to form 20 to 30 larger straight tubules called as tubule recti, which further forms the rete testis. The seminiferous tubular compartment contains the developing spermatids and Sertoli cells. Now, let us see the functions of Sertoli cells. Under electron microscope, you will be able to see Sertoli cells that appears as slender cells having irregular pyramidal shape. These cells perform important role, and these are: Sertoli cells provide physical support to the growing germ cells inside the testis. They nourish the spermatids and also remove waste products from the germ cells. They serve phagocytic functions too, that is, they eat away the cytoplasmic waste products which are formed during the process of spermatogenesis. They form a blood-testis barrier by virtue of their tight junctions. This barrier protects the germ cells from blood-borne toxins and antibodies. The barrier also prevents the entry of byproducts of gametogenesis into the blood circulation. It is because of this blood-testis barrier that autoantibodies against the germ cells are not developed during gametogenesis. And lastly, Sertoli cells secrete many important hormones and substances like inhibin, androgen binding protein, and estrogen. The second compartment of testis, that is the interstitial compartment, and it forms about 10 to 20% of the total volume of the testis. This interstitial compartments are present in between the seminiferous tubular compartments and consist of loose connective tissue and Leydig cells. Leydig cells serve an important function in male reproductive system That is, they secrete the male hormone testosterone. The two major functions of testes are spermatogenesis and endocrine functions, about which I will discuss in my next lecture. With this, I finish today's class. Thank you.